Hey Biochem, so I've decided to move this to a two-part video. So this very first video is going to describe about membrane protein structure and a little bit about their functions, but mostly focusing on their structure. So from this video, you should be able to describe membrane protein structure and function and identify and describe the association of membrane proteins to the lipid bilayer. And then there's going to be a second video that goes over a little bit more detail about selective binding and how to find things and interpret data quantitatively. Okay, so membranes are more than just lipid bilayers. Membranes also contain proteins and other biomolecules. So the phospholipid bilayer is the primary component of a membrane, but inside the membranes and outside the membranes, there's other biomolecules. There's carbohydrates. Those usually are on the outside of the membrane, and their function is primarily for signaling. So it would make sense that they're outside of the cell so they can interact with other cells or other molecules on the outside. The things that carbohydrates are attached to are either proteins or lipids. So if a carbohydrate is attached to a protein, let's see, here we go, attached to a protein, that protein is now called a glycoprotein. And if it's attached to a lipid, the lipid's called a glycolipid. So that also means that inside of the membrane and outside of the membrane are proteins. Okay. You probably heard of the fluid mosaic model of membranes, and the fluid part just refers to the membrane being sort of liquid or gel-like. If you remember, the phospholipids are very, very dynamic. They're moving sideways and they flip across. And so the membrane itself has like a fluid or gel-like feature to it. And then the mosaic part refers to all the other components that are part of the membrane. So it's not just lipid bilayer. There's lots of interesting things that are part of the membrane. And so we are going to focus on membrane proteins today. So proteins that are part of the membrane or associated with the membrane. Okay, and this is our last of the three different types of proteins categories based on their structure. So we've done globular, which are the water-soluble proteins, hydrophilic on the outside, hydrophobic on the inside, fibrous proteins that form insoluble protein fibers, and then today we are doing membrane proteins, and specifically we're going to look at one type of membrane proteins that are receptors. The membrane proteins, if they're associated inside of a membrane, like this structure here, they would be relatively water insoluble. And they're hydrophobic on the outside with a hydrophilic core, so kind of like the invert of a globular protein. And this feature of them is really important because a lot of membrane proteins have to allow through ions or molecules that are hydrophilic. They would not otherwise be able to cross the membrane. So the inside of the protein has lots of positive and negative charges and OHs for hydrogen bonding, but I wouldn't say all, but the majority of the hydrophilic amino acids are in the inside. And that means that the hydrophobic ones are out here, sticking out of the outside of the protein and having favorable non-covalent interactions with the lipid bilayer. So you can imagine some valines or some leucines or some isoleucines that are having favorable van der Waals with the inside of the lipid bilayer. Okay, so how proteins associate with the membrane gives them sort of their name. Okay, so we'll kind of go top and around. So these proteins up at the top here are called peripheral proteins because they hang out on the periphery of the lipid bilayer, or they hang out on the outside, and they associate or interact with the lipid bilayer with electrostatic interactions. These are also known as the non-covalent hydrophilic interactions. So these proteins are typically globular in that they have a hydrophobic core, but on the outside of them, like this one here, has some negative charges so that it favorably interacts here with salt bridging. And then if you imagine that these head groups here are negatively charged phospho head groups, that means that this peripheral protein likely has a lot of positive charges on it. Oh, I've zoomed in too far. Positive, positive, positive. So it can interact favorably with the phospholipid head groups. These could also be ion dipoles, salt bridge we just said, or H bonds, are how the peripheral membrane proteins interact with the periphery or the outside of the lipid bilayer. Another way that proteins can associate with the lipid bilayer are through what's called a lipid link, lipid linked protein. And these proteins will form a covalent bond with a fatty acyl tail. And that fatty acyl tail is what's associated into the membrane, and so the protein sort of just, dangling on the outside, but very strongly associated with that membrane. But doesn't make, doesn't make like full contact with the lipid bilayer except for through the acyl tails. Okay, and then the one that we're going to talk about for the vast majority is the integral membrane protein. That would be this one here. So the integral membrane protein spans the entire lipid bilayer. I'm just going to call it the entire bilayer. And these are the ones that I was talking about where the outside has to be hydrophobic to interact with the tails favorably, but the inside core is hydrophilic. And so how they're associated with the membrane is through mostly hydrophobic interactions. Okay, so what this image is trying to show is that if you had a detergent or soap, which remember soap is just fatty acids, you could pull the integral membrane protein out of the membrane itself because all of the fatty acid tails will favorably interact with the outside of that protein and like pull it out of its membrane. Okay, so like I said, we're going to focus on the integral membrane proteins from here forward because we're going to talk about receptors that are embedded in a membrane, and those are all integral membrane proteins. Okay, so the integral membrane proteins have there's two main possible structural features. The protein can be a bundle of alpha helices. 7 alpha helices is the most frequent. So if I looked at this integral membrane protein and the part that spans inside of the membrane, which the membrane here is supposed to be colored gray, so you can imagine that the membrane is here. The part that spans the membrane is called the I didn't fully write it out. It's called the transmembrane domain. Transmembrane domain. Okay, so the 7 helices, this would be like helix 1, 2, 3, 4, 5, 6, 7. It's debatable over whether this one is counted as helix number 8. Sometimes it's inside the membrane and sometimes it's out. Sometimes you include it as part of the other ones. Anyways, that 8th one is typically seen as being outside of the membrane, so it's not included as like part of the membrane. But anyways, you'll see 7 helices together in this particular shape, sort of like make a big giant cylinder around one another. Or you'll see a beta barrel. So it's about 16 to 18 strands, and this view has it when you rotate it 90 degrees, so you're looking down the barrel. You can see that there's like an empty space on the inside, so that things can pass through as a channel. And then if you wanted to label the N and C termini, there is a fun way of doing this. So when the protein is rendered like this in a ribbon diagram, where it has all the colors of the rainbow, so it's the visible spectrum, the N termini is where the blue starts, because blue is the CPK coloring for nitrogen. And the C terminus is in red because the C terminus has the oxygens on it, and red is the CPK coloring for oxygen. So I could trace the path of this protein, blue, and then it goes in the order of the electromagnetic spectrum. So 4 to up to green, to yellow, to the back, to the back, to the side. So the highest level of protein structure for this protein that's here is tertiary. It has just one subunit. And then I can also trace this beta barrel start to finish. Again, I would start with the blue, which you can't really see. It's like on the inside. And then I'd finish at the red. And again, this one has tertiary structure. So again, the integral membrane proteins typically have 2 structures. They're either mostly alpha helical or they're mostly beta barrels, and they tend to form these channels on the inside that are relatively hydrophilic on the inside, but they're hydrophobic on the outside. So like I mentioned before, we're going to focus on the integral membrane proteins that are also receptors. And receptors are proteins that bind ligands. So we've seen some receptors that are globular, like the human serum albumin is a receptor that binds to ligands, but that protein travels through the bloodstream. These receptors are going to be integral membrane receptors, so they're bound into the membrane. Okay, so we're going to talk through some of the vocab, but I'm going to draw a little bit of this out here. So if you imagine that I draw just a really blobby cartoon shape of a receptor, and I'll put a lipid bilayer around the outside of it. Just my phospho head groups. Tails. Hopefully you can see that there's 2 layers because it's a lipid bilayer. Okay. Tail. They all get 2 tails because of that glycerol backbone. Okay, the inside of this is hydrophilic. So if the inside of this could transport an ion, let's make it green. Let's say these are my ions. These could be sodium ions. if they can transport it all the way to the inside, then this receptor is called ionotropic. So this receptor would be ionotropic because it moves ions. Now, the way in which it moves ions is dependent on it binding to a ligand. So on the outside of the receptor, there are these regions where a molecule called a ligand can bind. I'll just make my ligand a circle for now. Okay, so when the ligand binds here, it can cause an allosteric change or a shape change that allows a lot of ions through. It could also cause a shape change to block the movement of the ions through the channel. And because there's a movement of ions that is regulated by a ligand, these ionotropics are also called ligand-gated receptor, because essentially the ligand is what's opening the gate or closing the gate to allow the flow of ions in or to stop the flow of ions in. And then again, just so that we're clear on our terminology, the protein here is called the receptor. So again, I could write this in a chemical equation, and I could have my receptor binding to ligand, and that would be in a chemical equilibrium with receptor bound to ligand. If it takes two ligands to bind, then I'd put a little subscript here and then 2 here. So there's an equilibrium. The ligand can come on to the receptor and it can come off of the receptor, and it's just very weakly bound through non-covalent interactions because you do not want a ligand permanently bound to a receptor. Okay, another type of integral membrane protein receptor is called a metabotropic or metabotropic receptor. Okay, and here I'm going to draw my receptor so that it has an open ligand cave or a place to bind it, but no tunnel going through it. Receptor. That's a protein. That's an integral membrane protein. So I'll just add my membrane. Bilayer, bilayer, tails, tails. These ones have other unique features. So typically, these metabotropic ones, receptors, are also bound to other proteins, like a G protein. And in this case, when the ligand binds, there's a change in shape that causes this G protein to release another receptor or another integral membrane protein to add some more membrane here. Come back to that G protein in just a second. Okay, so the G protein can release after the ligand binds, and it can go and bind to another protein receptor that could cause a change in shape here that lets ions come in. Let's say these are chloride ions this time. So that's one possible thing. It's possible that the G protein could activate an enzyme that then creates second messengers, and essentially all throughout the cell, other enzymes are activated, and there's a signal that gets propagated all the way through the cell. If the protein receptor is such a receptor, it usually causes overall a change in the metabolism happening inside of the cell. But then this receptor is called a G protein coupled receptor. GPCR is the abbreviation for this type of protein, the receptor. So the receptor is here. The G protein is attached to it. So just as a note, the G protein is not the receptor. It just happens to be attached to the receptor. So the two types of receptors that we have talked about now, the ionotropic and the metabotropic, are really common drug targets because the regulation of these can cause a variety of effects. So a lot of the drug molecules, and I'll just make, where should I put it? I'll make them in orange. Don't know why I picked the highlighter. Let's go back. A lot of the drug molecules that you may ingest target these receptors. And when they do target the receptors, there's sort of two very general possible things that happen. Okay, if the drug molecule gets there and binds, so in this case, the regular ligand is not there. If the drug molecule binds and upregulates the activity, it's called an agonist. So drug binding to receptor increases or upregulates its natural activity. Okay? If instead the drug molecule binds, let's just make a different one, maybe blue. A different drug molecule binds, so it's bound instead, and it shuts down the activity, is called an antagonist. So drug binding to receptor decreases the effect or the overall activity. And again, this can happen with the ligand-gated channel receptors or the G protein coupled receptors. This is just some schematic overview of a general case of, in this case, the extracellular signal, which is called a ligand, binding to a receptor protein, which is an integral membrane protein here. And then there's a whole bunch of things that happen inside the cell. And then here are some possible outcomes of, I don't know, what the ligand binding to the receptor. If you'll note, the ligand does, in this case, not enter the cell. It just causes a signaling cascade. So this whole thing is called signaling. And it's usually called a cascade because it's sort of like a domino effect. Once this one gets activated, then this one tips over, then this one tips over, and then just so many possible things can happen. It's like molecular dominoes. We also call this series of arrows where it's this happens, and then this happens, and then this happens, and then this happens, is called a pathway. There are more than just the two. There's more than just G protein coupled. Let's pick a highlighter. There's more than just G protein coupled and gated ion channel or ligand gated channel type of receptors. These are just the two most common targets for drugs, especially the ones that we're gonna look at. There are 4 other types. And if you'll notice, they all bind a ligand, which is an L here. And then they will cause some kind of signaling cascade inside of the cell. So another kind is a tyrosine kinase, and what to note about this one is that the inside part of this is an enzyme. So once the ligand bind, it activates the enzymatic portion of the receptor. Another type is called a guanyl cyclase, and again, when the ligand binds here, it activates a particular type of enzyme which causes GTP to be converted to cyclic GMP. Another type is the ligand passes all the way through into the nucleus and acts as a helper for transcription factors to turn on transcription, which then activates translation, and so the final impact of this is increased expression of a particular type of protein. Okay, and then so before we move on to part 2, I just want to make sure we're all super clear that this is the receptor protein, which is integral membrane protein. This is also a receptor protein, which has an integral membrane or transmembrane domain. This one is also an integral membrane protein with its transmembrane domain right here. And this one right here is also integral membrane protein with the transmembrane domain. This flavor and this flavor do not follow the integral membrane structure function type of situations. So we are going to move into part 2. So now would be a good time to take a break, make sure you've got all your notes ready, and then we will get started on the next part. Okay, so the second part of this is